Good morning, all you cats and kittens. Um, Welcome back from spring break. Today, I'm going to take you through, we're going back to genetics. We're going to learn about dihybrid crosses today. You took notes over them before break. Now we're going to learn how to actually do a dihybrid cross. All right, so let's kind of look back at what we did before break and then pick up some new skills today. So before break, we did something called a monohybrid cross. All right, and we're going to start with comparing that to a new skill called a dihybrid cross. Okay, so a few things about monohybrid crosses. We used the monohybrid cross when we looked at just one trait. For example, maybe eye color. Looks like an L trait, so I'm going to change that to a 1. One trait. It looked at one allele, so we only used one type of letter. Maybe we used B's. It could have been uppercase or lowercase, but just one letter type because we only had one trait. And those crosses were done in what's called a Punnett square, and we used just 4 boxes. Like that. Okay. A dihybrid cross you will use when you are looking at 2 traits. So let's say we want to see what happens with height and eye color, right? Someone's tall with brown eyes. How's that cross going to go? We're going to use 2 alleles. Right, so for height, maybe we're going to use T's, and for eye color, we're going to use B's. So maybe we have someone that looks like that and someone who looks like that, or this could be the same person. They could be tall with brown eyes, but heterozygous for both traits. And the dihybrid crosses use 16 boxes. Okay, so it's much bigger Punnett square. Alrighty, so let's do a monohybrid cross just to refreshen our memory, and then we'll work on, first we'll just show you what a dihybrid looks like, and then we're going to write down all the steps on how we did this cross. All right, so for a monohybrid cross, let's say we have a homozygous dominant brown-eyed person crossed with a homozygous recessive blue-eyed person. And so we draw our classic 4-box Punnett square. Because of the laws of segregation, the genotypes, the alleles and the genotypes segregate, they separate, right, and they form the sex cells, right? This is one sperm cell, this is an egg cell. So this person made sperm, this person made eggs. And we're only going to put one of our alleles from our personal genotype, our diploid genotype, is going to go into our haploid sex cells. All right. Then we recombine. All right, and all of the offspring end up being heterozygous. All of the offspring would express brown eyes, so they would have the phenotype of brown eyes, but they would all be heterozygous. So they could go on to have children with blue eyes possibly. Okay, so let's do a dihybrid cross now. So let's say we have a person that is heterozygous, tall, heterozygous, brown eyes. So this is person 1. Okay, and we're going to cross them with person 2, who's homozygous, dominant, tall, homozygous, dominant, brown eyes. So this is person 2. Okay, so I'm just going to show you what this dihybrid cross would look like. You'd want to give yourself a lot of space. It's going to have to be much bigger than what this 4-box was. 1, 2, 3, right? 3 lines in the middle if we want 4 columns. 3 lines, 2, 3 lines again. I'm just going to put, we're going to put letters on the outside just like we did for a monohybrid cross. There's a certain way to do that. I'm just going to show you what it looks like right now, and then we'll break down the steps of how I got there. So the alleles are going to segregate, right? So this is going to be a sperm cell, sperm cell, sperm cell, sperm cell. In this case, we're looking at two different traits carried in each sperm cell. So this is one sperm cell. This could be another possible sperm from this person. This could be another possible sperm from that person. Or this could be a possible sperm from that person. All right, so let's talk about the eggs. The eggs are going to be less diverse because they're homozygous dominant, homozygous dominant. So every single egg is going to be big T, big B, big T, big B, big T, big B, big T, big B. All right, so that's how we set it up. And then what it looks like to recombine these, we're going to put the T's together, just like we were doing a monohybrid cross. So drop and drag over. And then we put the B's together. So we don't stagger them. We don't go TB, TB. We go TTBB. Okay, so for this one, same thing. The T's come together, and then the B's come together. So you'll see what we're forming as these are the diploid offspring. These are the possibilities of the offspring. So we're haploid gametes on the outside, diploid offsprings, just like the people were at the top. Okay, so that's as much as I'm going to show you on a dihybrid up here. We're going to now break down the steps for solving dihybrid problems. Okay, so let's write that. Steps for solving dihybrid problems. All right, we're going to say step 1. Read the question and assign alleles. All right, so you have to figure out what the genotypes of the people or the plants or the animals in the possible question are. So up here, this person was heterozygous for tall, heterozygous for brown eyes. So maybe the problem, it doesn't give you the letters. You have to choose, okay, I'm going to use T's because we're talking about tall. I'm going to use B's because we're talking about brown and blue. So you just have to kind of come up with that yourself and assign the genotypes. But that was something you had to do for monohybrid crosses. So that's not going away, that skill. You'll get more practice with that. All right. That's easy. That's something we've done before. Step 2, this is where, hang with me, folks. We're going to do something called, we're going to foil the genotypes. So what this is going to do is this is how we have to foil the genotypes to go from a diploid genotype to a haploid gamete or sex cell. We have to foil this. All right, well, what does FOIL stand for? FOIL stands for first, outer, inner, and last. Okay, so let's foil the genotype. The first genotype we're going to foil is person 1. So heterozygous for tall, heterozygous for brown. Okay, I'm going to use some different colors to help us keep this stuff straight. All right, so when I say foil, we're going to start with the first. That means the first letter of each genotype. That would be the big T and the big B. Okay, so that's our first foiling, big T, big B. And that's what I'd already put up here. Okay, the next one, I'm going to switch pens again. The next one are our outer. Those are our letters that are on the outside of the genotype. So in that case, it would be this big T and the little b, outer. big T, little b. I'm going to draw that loop for the, okay. All right, then we're going to move to inner. That means those are the letters on the inside of the genotype. The inside of the genotype, so that's going to be this little t and this big B. So it's the inside guys, okay? So we've got a little t and a big B. You probably think I'm a big B, but hey. All right, and then we're going to do the last letters of each genotype. That would be the little t and the little b. Okay? So that is foiling. And if you check the problem, I kind of started up here, you'll notice that's what I can do it in my head. You know, hey, there she is bragging again. But you'll be able to do that at some point too. So this will be something you can just kind of look at and do by, anyway, so that's what I did up there. So now we're going to foil the next genotype, which was homozygous T, homozygous B, dominant for both. So let's write that genotype. Let's write out our foil. Okay, and then let's write our genotype. Okay, and let's do the same thing. We're going to switch lots of pens. Start with red. We're going to do first. That's the first letter of each genotype. All right, outer. That would be the outside letters of the genotype. Inner would be these inside guys. And then last would be the last letter of each genotype. Okay, and you're probably smarter than me, so you should probably have noticed that every single one of these were going to be exactly the same. Because if we're crossing two homozygous, then all of the foiled products are identical. And if you look back up here to our example one, they were all big T, big B. All right, so we've foiled our genotypes. All right, next step. Okay. Next step. We're going to do step 3. We are going to write foiled products, those gametes we produced, on the outside of a 16-box Punnett square. Give yourself space. You can never have too much space for a dihybrid because you have to put these letters back together. So you always want to make a big box. Okay, looks pretty good. Now we are going to put our foiled products on the outside of our box. So I'm going to keep the color coding. So this was person 1. This was the first one we foiled together. And that was the one that had the most diverse foiling products. The next person we did had the exact same foiling products. They were dominant for everything. So just one more time, I got these products, right? I just copied what we did over here. So this was person one. I put them on the top. These were the products I put on the top. This was, all right, this was person one, and this was person two. Okay, and then I put person two products on the side, consistent with what I'd already done up here, but I'm just breaking it down for you step by step. Okay, the fun part. Step 4, we are going to combine alleles in the boxes to create the possible genotypes of the offspring. So if these two people would get together, this is what their children could possibly turn out like. Okay, so let's do that. We're going to combine these alleles. We're going to keep our T's together and our B's together. You need to treat this just like you would a monohybrid cross, right? You put the capital first and then the lowercase, and then you keep your letters together. Don't stagger them. So what does that mean? That means we put the two big T's together, and then we put the two big B's together, right? This box, we're going to have big T, big B, little b. Big T, big B, big B. And then we have big T, big B, little b. So that's our first row. So we just combine the letters back together, and you'll see we have haploid gametes on the outside. We've got haploid sperm, haploid eggs, and then they come together to make a diploid person. So, alrighty, so let's just keep going. Big T, big B, big B. Big T, big B, little b. Big T, big B, big B. Big T, big B, little b. Voila. All right, we got that. So now we're going to do the last step. Hey, we've made it to the end. Step 5. You need to analyze the results, right? The question, they're going to ask you a question about this Punnett square. So let's go through what our alleles mean. So we were looking at the traits for height. So we're going to say big T is tall, little t is short, big B is brown, little b is blue. Okay, so looking at this isn't the most interesting of results because every single possible outcome has at least one capital letter. And so that means they're going to have the dominant traits. So that means 100% of offspring will be tall with brown eyes. So not that interesting of results, but you know, hey, it is what it is. And that is how you do a dihybrid. I have no idea how long this video is right now. Okay, we're just at 20 minutes. I can keep you on the hook. All right, so that's the notes part. I'm going to do one of these practice problems that you're assigned. So we're going to do problem A. It says, some flowers are dominant to short, while yellow colored flowers are dominant to orange. Cross a plant that is heterozygous for both traits with a plant that is heterozygous for height, but homozygous dominant for flower color. Okay, so the first thing we have to do is come up with genotypes. So let's assign alleles first. So we have to assign alleles. So we're doing tall and short. So let's just keep with, so this is, let me change, practice problem A. Okay. Page 9 of your packet. All right, so let's do big T for tall, and we'll do little t for short. Okay. And then the next, we're talking about flower color, yellow and orange. And I actually don't like to use Y's or O's because the uppercase version of both letter looks a lot like the lowercase version of those letters. So I don't like that. So we're going to use A's instead. So if this is confusing to you, then go ahead and use Y's. But I guess we could write it like that maybe. Let's give it a go. And we're not going to use A's. Let's try Y's. I'm afraid to just throw you off too much. So this will be yellow, and then we'll do a loopy Y for lowercase, and that will be orange. So I guess those aren't so bad. But imagine if we used O's, right? That would be uppercase, orange, or I don't know. So some letters are better to use than others, but we'll use Y's. Why not? All right. So now we have the alleles we're going to use. We can assign genotypes. So we are going to cross a plant that is heterozygous for both traits. All right. So we're going to have plant 1 is heterozygous tall, heterozygous yellow. I'm already sucking on the loopy Y. Loopy Y, there we go. Okay, and then we'll have plant 2 with a plant that is heterozygous for height, but homozygous dominant for flower color. All right, heterozygous for height, so that's a big T, little t, and then homozygous dominant for flower color. All right, so those are the genotypes we're working with. Now remember, we have to FOIL both of these genotypes before we can even put them on a problem. So let's FOIL it. These are going to give our haploid gametes, our sex. So this is our potential sperm or potential eggs that we could create. So remember, first is the first letter of each genotype. So big T, big Y. And for this parent, it's also a big T, big Y. All right. Okay, so we're going to green. All right, next is the outer. So that's the outside letters. So for this, it's a big T and a lowercase y. And over here, it is a big T, big Y again. All right. Then we're going to do inside. So that's these little guys or big guys in the center. So we have a little t, capital Y. And over here, same thing. We got a little t, capital Y. Yay. And then the last one are the last. So that's the last letter of each genotype. So we got little t, little y, little t, big Y. Okay, so that's fun. So now we're going to have to draw the big square, or if you're using the document, you can type into it. I don't care, like always. Do it by hand, do it on the computer. I'm a by hand kind of person, but maybe you've printed this off because you're one of those cool people with a printer, or maybe you're like me and you don't have a printer, so you just write everything by hand. And by the way, also not having a printer is also makes you a very cool person. So not just the printer people, despite how they constantly brag. You know you do. Okay, so let's put these phenotypes on the outside. We've got big T, big Y. So we're going to put plant 1. Then we got big T, little y. All right, and then we've got little t, big Y. And finally, we got little t, little y. And then on this side, we've got plant 2. We're going to do big T, big Y. Big T, big Y. Little t, big Y. And little t, big Y. Okay. So now we're going to put these suckers together. Okay, I'm just reread the problem, made sure I, didn't mess up. All right, here we go. So put the T's together, and then put the Y's together. T's together, then put the Y's together. Oh my gosh, I forgot what I was doing. Okay, my brain keeps like popping in and out of consciousness. Oh my goodness. Okay, we got that. Let's do this one. T. Are we having fun yet? You all said yes. I'm sure you did. Because I'm having fun. I love talking to a computer. No one to respond. Just great. Oh, I miss your little giggles or your eye rolls. I'll give anything to see you with your screens out right now, ignoring my lecture. All right. We did it. We put them together. We did it. Oh my God, we're at 28 minutes. Okay. So let's see. Phenotypic ratio. Add text here. Okay. So let's figure out what we just got from this cross. Okay, so our possible options, we have 4 possible outcomes. We have plants that could be tall and yellow. We could have tall and orange. We could have short and yellow, or we could have short and orange. Okay, so to be tall, it would have to have at least one dominant T. It could be homozygous dominant or it could be heterozygous. And the same thing for yellow. It has to have at least one big Y. So let's look at all of these and let's figure out how many have a big T and how many have a big Y. All right, so we've got this one, big T, big Y. We got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. No, 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 no. All right, so we got 12 out of 16. And if you break that sucker down, you get 3/4 or 75%. Okay? How many are tall orange? That means we would have gotten big T's and lowercase y's. 2 lowercase y's. And our answer to that one would be 0 out of 16. I don't know why I couldn't write a slash, but hey, struggle is real. So 0% are tall and orange. Okay, how many plants are short and yellow? So short would have to have lowercase t's, yellow would have to have at least one uppercase y. So we've got lowercase t's, and an uppercase y, lowercase t, uppercase y, lowercase t, uppercase y, lowercase t. Okay, so we got 4 out of 16 are short and yellow. So that would be 1/4, which would be 25%. Okay, and then how many short orange? We had 0 out of 16, 0%. We got no orange flowers, but we didn't have any orange to begin with, right? Both of our parent flowers were yellow. Okay, so that's a fairly large video file. So we're at 32 minutes. So I'm going to stop there. You need to finish the rest of this sheet. So get your little brains working. Reach out if you need help. If I get a lot of people reaching out, I can make a second video. I'd like you to try them on your own, or maybe I'll just post the answers and you can grade yourself. But if you need another video, I'm happy to do it. Well, I'm not happy, but I'll definitely do it. All right, bye. Miss you guys.